So after we have looked into oxytocin, we now move on to the second video on vasopressin. Vasopressin was also known as ADH, antidiuretic hormone. In Chinese term, it's called 抗利尿激素. Okay, and vasopressin, vaso means the blood vessels. Okay, so that would pressin mean to depress. And then, so the action is actually related to the kidney function for water retention. And it would cause arterial constriction because when this hormone, ADH, acts on the V1 receptor, will bring about the increase of blood pressure. So it's also known as 加壓素. Increase water intake to produce the sensation of press. This would bring about the feeling of getting more water. So if you become thirsty, perhaps it's induced by vasopressin. And overall effect is to reduce plasma osmolarity. So in other words, this hormone would control or modulate blood plasma osmolarity. Okay? Because as the blood osmolarity increases, the arginine vasopressin would increase as well. That would bring in for more water retention, and the end result is to reduce the blood osmolarity. We take a look at this in further details. Now, similar to oxytocin, is actually coming down from the hypothalamus, from the neuron or the neurohypophysis, to release vasopressin. So this vasopressin would go into the kidney, and the kidney, it would act on V2 receptor, and then that would bring about the retention of water. So more water would come in. So the decrease of plasma osmolarity would end up getting down into the feedback here to inhibit the release of vasopressin. So vasopressin or ADH is induced by drop in oxygen level in blood. And then there are also other hormones like angiotensin 2. We'll discuss about this on in video 3. Adrenaline and also cortisol. And of course it's related to stress. All right. So the increase in plasma osmolarity would induce the release of this vasopressin, and then end up with the decrease of plasma osmolarity, and then with that would inhibit the more vasopressin would have a feedback loop to inhibit all this, and then this would bring about the balance of plasma osmolarity. So you can see this hormone is related to plasma osmolarity. You can see on the right hand side here, the plasma vasopressin level would go up, and then that would, now this, pay attention, this slide is on the urine osmolarity. So the urine osmolarity would be increased, okay, because that would be bring about the water retention, so there would be more water. And then with the more water, the urine osmolarity would increase as well. And then the relationship of plasma osmolarity and vasopressin levels is shown in this diagram. Actually, these two diagrams taken from the textbook, but as you can see, the plasma osmolarity that is in the blood, and then that would increase, and then you can see the vasopressin level also increase. So they have a positive correlation. And then you have more urine osmolarity resulting with increased plasma vasopressin. Okay, so we need to control the water content in our blood. And then how? Okay, so we need the kidney. Normally, kidney is to maintain the body's water content, because the next slide will show to you in the actions, the signal to release ADH or 抗利尿激素 or AVP will bring about the contraction and relaxation of small muscle and promote water moving across the epithelial cells. Okay, And these would be related to its action in the kidney. We'll talk about it in the next slide. But before that, because of the control of the water content, blood osmolarity is also related. The blood, we just examine that, the positive correlation. The blood osmolarity increase, and then more AVP release. And then the AVP will control the blood level. And also with the decreased blood volume and blood pressure, when it's decreased, it will increase the AVP release. The net result would be the action of this hormone to act on the AVP-V2 receptor, coupling with the Danner cyclase for the antidiuretic effects. And it's also acting on the arterial and also the liver V1A receptor, mobilize calcium signaling, and that would bring about the control of the release for water and iron retention. And then in the brain, the adenohypophysis will have type, and this would trigger calcium ions to release ACTH. Okay, so that's why we put ACTH in this lecture here. Let's take a look at what's going on in the kidney. In the kidney, it's actually controlled by this AVP secretion and control mechanism. So the AVP would increase if it's stimulated by nausea, vomiting, stress, and exercise. So these actions also bring about the release of ACTH from the anterior loop. And then for AVP increase, it turns into smooth muscle, vasoconstriction, move into the kidney. That will stimulate what we call this is the water pump, aquaporins. And then aquaporin increase the action that would bring about water retention. At the same time, ACTH would go into the kidney, and that's the kidney. And that would turn on aldosterone with the action of the cortisol or aldosterone, will stimulate sodium and water retention all together. So it is also related to blood volume. When the blood volume decrease or the osmolarity increase higher than these 280 milliosmol per kilogram, and that will bring about the stimulation of the release of AVP, vasopressin, and also ACTH. So in other words, AVP could stimulate ACTH as well, and potentiate CRH. CRH is from the hypothalamus, corticotrophin releasing hormone, on the pituitary gland to produce this ACTH. And then focusing on the kidney, this is the structure of the kidney's epithelial cells. This is the apical side, this is the basal side. And then these are the structures of the aquaporin, the location of the aquaporin 2. And the aquaporin 2 is on the apical side, 3 is on the basal side. As you can see that, the hormone will stimulate cyclic AMP, bring about the formation of more AQP2, aquaporin 2, to bring in water, and then at the same time stimulate more AQP3 at the basal side to bring about the retention of water and going back into our body. Okay, so AQP again is a water channel. 1 is responsible for renal water retention in the proximal tubules. Here is the epithelial cell. 2 is in the collecting duct at the apical side, and then cyclic AMP signal given by these hormonal effects on cyclic AMP will stimulate this to bring in more water. Aquaporin 3 at the basal side also activated by this hormone action on cyclic AMP to bring in more water into our body. You may be interested in knowing by studying those diseases. This is one very rare inheritable disorder or genetic disease known as autosomal dominant neurohypophysis diabetes insipidus, or ADNDI. ADNDI. Okay. The symptoms of these patients will have polyuria. Poly means more, urea means more urine. The patient will have more than 2.5 liters per day of urine. Normal person, I'm not so sure whether you realize or not, perhaps would be like, for example, less than a liter or half liter. So this is about 2-3 times more water. So when you have more water going out, you will become more thirsty. So therefore, we have polydipsia, meaning excessive thirst, and then leading to what we call dehydration. Okay, that because it's a genetic disease, so this thing would happen in the childhood. It's actually found to be related to the mutation of a gene. The gene mutations actually are found in the neurophysin 2 because it's in the AVP and MP2 gene. So neurophysin now later on is known as the binding protein for arginine vasopressin. The mutation is actually on this location of the cysteine. So what's the importance of the cysteine residues? As you can see, this is the normal allele and then mutant allele. From cysteine mutated with PON mutation to phenylalanine. Okay, so without this cysteine, so this is the second, the tertiary structure or the model of neurophysin. Neurophysin actually would work. They would bind to arginine vasopressin, and they form a tetramer with this dipeptide. And this dipeptide formation, this is the arginine vasopressin, and this is the location for these two peptides forming together. The formation of the peptide actually require disulfide bonding. So therefore, without the cysteine, they don't form this tetramer. Okay, this is the normal type. Okay, with this dipeptide bound to it. Okay, without this tetramer, they cannot bind to the hormone. But that is to protect the hormone from being degraded. So the plasma hyperosmolarity and accumulation of mutated protein. So the mutated protein actually could not be functioning. So you keep on producing this protein, and then the protein would cause apoptosis of neural cells, and more severe problems actually is found in the brain of these patients. Okay, so this also demonstrates the importance of this NP2 or neurophysin 2 in binding with the hormone. Okay, and let's move on to the final story of this video 2. We are trying to explain the actions of this hormone, so we would like to understand the two hormones. They are very similar. They are just like brothers or cousins. And then how about their receptors? Their receptor actually, they are all G protein coupled receptors with 7 transmembrane domain, but they have some fundamental differences, perhaps mainly in the extracellular space here, or sometimes we call this the docking site of the hormone. Okay, so they have a number of key features, including they are G protein coupled receptors, and then the oxytocin or arginine vasopressin would bind to the N-terminal or the docking site. And then some parts of the transmembrane domain, would also affect the binding as well. There are also arrestin binding, which is a peptide or protein to arrest or to stop the reactions of this receptor, and also phosphorylation would form the what we call the desensitization. And then after that would be internalized by endocytosis to remove the whole signal. Okay, so they're very common features. but actually they are from two different genes, not located at the same chromosome locations. Arginine vasopressin receptor 1A is actually on chromosome 12. It has a structure like this, the exon, and then the other exon, very small exon numbers with a big protein. And there are also many differences, like for example, the regulatory element here is with the GATA, and then here is the structure of oxytocin receptor. It's located in chromosome 3. And then you can see that on this chromosome, the intron here is very long. There are a couple of exons, and then the coding region is in purple. And then you can find more gene mutations in the intron. Okay. So the docking sites for the oxytocins are here, and then this is arginine vasopressin. So the docking sites actually are also here. So in other words, the two receptors have similar structures, but their genes are very different and not linked together, not at the same position in the chromosomes. They are also under different controls of the different mechanisms of gene regulation. There are mutation noticeable in the arginine vasopressin receptor 1A. There are insertions here with different lengths. So in the 5' franking region, but for the oxytocin receptor, the intron here would have several different mutations that would affect the function or the expression of the oxytocin receptors. Here's one example of these changes of the gene expression levels. We are talking about ABP receptor 1 gene. They have a long and also the short transcript. The long transcript I've just mentioned, those are microsatellite sites. These are the elements here. Sometimes they have shorter repeats, sometimes they have longer repeats. So the microsatellite, they have longer, they form this long transcript. Shorter, they form this short transcript. So there are what we call the genetic polymorphism on gene expression level and brain activation and social behavior. It is also known to be related, in this case, in human, the microsatellites here, and then the long and the short. You can see in human we may have two short, or one short, one long, or two long transcript. And then the relative gene expression level for the long form is higher than the short form. And the long form and the short form, they have different position on the gene expression in the brain, meaning that these receptors would have a different relationship to control the action of arginine vasopressin in discrete brain regions or in different brain regions. And this brain region is believed to be to affect social behavior. That was almost 10 years ago that Donaldson and Larry studied those ABP receptor 1A with two transcripts, one long one with longer microsatellite, the short one with shorter microsatellite in different animals or in human. And perhaps they have profound effects on human behaviors or animal behaviors. Okay, we'll come back to the details of this. Actually, you're required to do your term paper or term essay. This is one of the exercises that you are required to do. So I'm not going to give you all the details, but just to provide you with some basic background on ABP receptor 1A with a long form and a short form, and perhaps the different location in the brain would affect animal behavior, including human behavior. So the case studies for the genetic diseases relating to this receptors. Case number 1 is the CNDI. CNDI is congenital nephrogenic diabetes insipidus. So usually in the first week of life with vomiting, constipation means lack of water, okay? Failure to thrill, fever, and polyuria. Remember, polyuria means a lot of urine in the patients. So the patient have very low urine osmolarity, so because the volume is so big, iron has been diluted, so the osmolarity reduced, and high blood vasopressin level. So could you suggest possible causes of CNDI? You may click into the PDF file to look for the information about this case. Obviously, you could think of this is related to the mutations found in the vasopressin receptor that would bring about this polyuria, because the receptor perhaps is working or not working properly. Therefore, we cannot control the urine level, okay, leading to polyuria. Another case is known as excessive vasopressin, also with a high blood vasopressin level, but it's called syndrome of inappropriate antidiuretic hormone, or SIADH, with again also hypoosmolarity in patient with hyponatremia, mean very low sodium level. Suggests possible cause of this SIADH. Now in this case, one drug is approved to treat SIADH. It is ribosol. You may check on ribosol, clicking right click, you can search for the function of this drug. So obviously this drug again is to act on, on the receptors. Okay, and at the same time, could you suggest the drug how does it act on the receptor, or where does it act on, and the side effects that it could be made for this patient. Okay, you may again click into this review from an article about SIADH. So I'm not going to tell you all the answers here, but I've already given you some hints onto the drugs and also the patients relating to these genetic diseases. So in summary, what we have explored in this video, including video 1, AVP or arginine, vasopressin, and oxytocin are two neurohypophyseal hormones from the hypothalamic neuron. They act on their own specific neurophysics. NP1 for oxytocin and NP2 for arginine vasopressin. So AVP control osmoregulation by acting on the kidney to retain water retention. As a result, AVP and other factors work coordinated to control of blood pressure. And then we have AT2 receptor mimics that could help in controlling blood pressure. We also have oxytocin activates smooth muscle contraction by changing or facilitating the calcium release or even calcium intake in myoepithelial in the breast tissues, and also the myometrial cells in the uterine. So oxytocin works to promote bonding among humans. So that's why it's known as cuddle hormone. We have explained that previously, and then to remind you that oxytocin is not to be regarded as a love hormone, because the application of these drugs could have severe effects on us. And then the AVP receptors and also the OT receptor also plays a significant role in human social behavior for sure. Okay, so let's stop here for the second video.